So thank you so much for inviting me here to give this lecture. It's a great honor. And today I'll be talking about how we can investigate the causal effect of maternal and intrauterine exposures on offspring perinatal health and risk of future cardiometabolic disease. Using new statistical models in genetic epidemiology. So, as you said, was here in 2016. It's quite a few years ago now. Do I have a pointer? Nope. Oh, no, I don't have a pointer. That's all right. We can do it without the pointer. So yeah, I was a first-year PhD student when I was here. I'm quite in the middle, in the front, if anyone can find me. This year was, I would say, I didn't realize at the time, but I think changed my future career quite a bit, both in that I met Dave Evans here at the then pre-course in genetics, which is now the specialized course that will be next week, and also met Deborah, who's teaching in week 3, who's become a close collaborator together with Carolina. So this course has given me a lot. When I was here, I did my PhD in genetics and glucose metabolism during pregnancy and gestational diabetes. And through these encounters and meeting with these collaborators, that sort of developed into the space that I'm now working on with the developmental origin of health and disease hypothesis and looking at exposures in mom and offspring outcomes. So I guess that's enough down memory lane. We'll start talking about the science. So all of these hypotheses that I will be talking about today sort of started with David Barker and the thrifty phenotype hypothesis in the late 80s. So David Barker was a British epidemiologist who used historic cohort data to look at the relationship between low birth weight, premature birth, and growth retardation and later life disease such as hypertension and diabetes and coronary artery disease. And he suggested that there was some sort of intrauterine mechanism that was leading to both these birth outcomes and these later life diseases. One of paper I would like to highlight is this paper looking at the prevalence of impaired glucose tolerance or type 2 diabetes in 64-year-old men in the UK. And as we can see on the y-axis is the prevalence of type 2 diabetes, and on the y-axis is individual's birth weight. And as we can see, for those who were born with a very low birth weight, so that's less than 2.5 kilos, they had a significantly higher prevalence of type 2 diabetes. And as the birth weight got larger, the prevalence of diabetes seemed to go down. And so from the Barker hypothesis, the sort of developmental origin of health and disease hypothesis developed. And that hypothesis in general looks at exposures to certain environmental influences in utero and look at how they may have a significant outcome on an individual's both short and long-term health. So typical exposures that we look at is poor maternal nutrition, chemicals, infections, or stress. But there is potentially also another explanation for this relationship between low birth weight and later life disease. So here I just want to talk a little bit about the fetal insulin hypothesis. So this paper here was published by Hattersley and colleagues and looked at mutations in the glucokinase gene and birth weight. So this GCK mutation does so that the body senses glucose at a little bit of a higher level than normal. So individuals with this mutation have slightly higher glucose throughout their lives. And what they saw here studying 23 families was that if this mutation was present in mom, but not present in the fetus, the offspring was born with about 600 grams higher birth weight. And this is because when glucose crosses the placenta, the fetus senses this glucose at a higher level, producing more insulin, and insulin is a growth factor, which then causes this increased birth weight. If this mutation was present in both mom and the offspring, they didn't see a difference in birth weight compared to normal pregnancies. However, if this mutation was only present in the offspring, so inherited from dad, we saw a lower birth weight of about 500 grams, and this was because the fetus would get glucose at a normal level from mom, but sensing it at a different threshold and producing less insulin. So we can kind of look at this observed relationship between birth weight and later life disease two different ways. So the thrifty phenotype hypothesis sort of looks at this as more of an environmental explanation. So, for instance, poor maternal nutrition can lead to decreased supply of nutrients and then leading to lower fetal growth, resulting in lower birth weight. And this also leads to some sort of an intrauterine programming of the metabolism that will later in life increase that individual's risk of disease. Whereas the fetal insulin hypothesis suggests that it's gene influencing insulin secretion and action that causes lower fetal insulin-mediated growth and lower birth weight, and that those same genes also decreases the capacity to secrete and response to insulin, leading to an increased risk of disease in later life. And so the Barker hypothesis and the Doe-Had hypothesis, they all sort of build on these observational studies, but we do, of course, know that there is some potential problems with drawing inference from these studies. Things like reverse causation, confounding, and biases can be a problem. Perhaps maybe not reverse causation in this case, because we're looking at exposures in mom and outcomes in the offspring, but confounding, for instance, could be a big problem. For instance, if we think that we have an exposure, such as maternal smoking, that we know will increase offspring birth weight, that is likely to also increase the chances of the child started smoking when they get old, and we know smoking can cause many of these outcomes that we look at. So it might not be it's the intrauterine environment itself that's causing some sort of programming, but it could be other factors in the environment. And so a lot of the work that I've been doing since I was here at the course is to try to look into this relationship between intrauterine exposures and later life disease and try to work out whether that is actually causal. And so the gold standard when we look at causality is, of course, the randomized control trial, where we randomize individuals into some sort of a exposed group and giving them an intervention, and a control group that doesn't get this intervention. And we can then compare the outcomes between the groups, but this is not always ethical or practical, particularly looking at toxic exposures, smoking, alcohol, looking at things in pregnancy, so everything related to the Doe-Had hypothesis would be difficult to do in terms of an ethical standpoint. Also from a practical standpoint, we would sort of have to give someone an exposure or intervention in pregnancy and wait 50, 60 years to see if they develop any disease. So this option is sort of excluded when we want to look at questions related to the Doe-Had hypothesis. So then the question becomes, how can we address these potential causal effect of intrauterine environment on offspring outcomes? And one method that I will spend some time today on is Mendelian randomization. So we can draw some parallels between randomized controlled trials and Mendelian randomization. So where, as in an RCT, we would physically randomize individuals to an exposed group and a control group, we use the random segregation of alleles in Mendelian randomization to determine if individuals are in a sort of an exposed group that have some sort of functional allele or a control group. And we can then measure the outcomes between those two groups. As with any method, there are some assumptions with Mendelian randomization that we need to be aware of. So we need genetic variants or single nucleotide polymorphism or SNP that we know are associated with our exposure of interest. Typically, in the day and age now where we have big genome-wide association studies, we find our genetic variants in those kind of studies from the exposure of interest. We also want the SNP to not be associated with any confounding variables. So this should hold true due to the randomization of the alleles and the segregation. But, and we can test it to some sort of degree if we have measured confounders in our data set. But we all know that we don't always measure confounders perfectly, and we can't test this for confounders we don't know about. So it's somewhat testable, but not entirely. And thirdly, we need the SNP to only be associated with our outcome of interest through the exposures. There could be no other pathway in which this genetic variant is associated with our outcome of interest. This last assumption becomes tricky in many ways, and particularly when we want to look at this in a multi-generational design. So when we have a maternal genetic variant that is associated with our maternal exposure of interest, and we want to look at an outcome in the offspring. This is because we know genetic variants in mom are correlated with offspring genetics, and we know that offspring genetics can have an effect on their own outcomes. So when we want to investigate this Dohad hypothesis, we need to be careful to take offspring genetics into account in our analyses. So there are several ways we can do this. So we can perform Mendelian randomization in mother-offspring pairs or mother-father offspring trios, and doing a one sample Mendelian randomization where we have both genotypes, exposure information, and information on the outcome in the same sample. Or we can do a two sample Mendelian randomization of maternal exposures on offspring outcomes, and here we need the SNP outcome association to be adjusted for offspring genotype. So what we are interested in is this beta causal, so the relationship between the exposure and the outcome. From one data set, we can get this beta 1, so the relationship between our genetic variant and the exposure of interest. And from a second, typically a genome-wide association study, we can get our beta 2, so the association between our genetic variant and our outcome. And it's this association here which needs to be adjusted for offspring genotype. So before I go into talking about some of the work that we've been doing with Mendelian randomization, I want to talk a little bit about some genome-wide association studies of birth weight that have been performed, both because this is where we get our genetic instruments when we want to do Mendelian randomization between birth weight and later life disease, but also because there's been done a lot of important work looking at genetic correlations between birth weight and adult disease that I think is important to keep in mind. So firstly, I would like to talk about this egg consortium GWAS that was performed in 2016. So here is the Manhattan plot of this GWAS. So in this plot, every dot represents a genetic variant, and the x-axis is where that genetic variant is placed along the chromosomes. And on the y-axis, we have the minus log 10 p-value. So that means that the higher the tower, the more significant the genetic variant is associated with the trait, so birth weight in this case. And we can see here that they found quite a lot of genetic variants associated with birth weight. And one of the things that they did in this paper that I think quite interesting was that they looked at the genetic correlation between later life disease. So On the x-axis is a bunch of traits that they looked at, and on the y-axis is the genetic correlation coefficient between birth weight and these traits. And I just wanted to highlight the cardiovascular and glucose-related traits where they did see a quite strong negative correlation between an individual's birth weight and their risk of later life disease. So I guess this study suggests that there is a big genetic component, and that which also relates to the fetal insulin hypothesis, but it doesn't necessarily exclude the Doe-Had hypothesis or the Barker hypothesis. But one of the other things that they also looked at was how much of this observed association between birth weight and later life disease was due to shared genetics. They looked at this for type 2 diabetes and systolic blood pressure, and they saw that the proportion of covariance explained by genetic variants was as high as 0.96 for type 2 diabetes and 0.85 for systolic blood pressure, suggesting that quite a lot of this covariance that we see is due to shared genetics. However, as we saw from some of the work with the GCK mutation, an individual's own birth weight is not just a product of their own genetics. Mom's genetics is also quite important when it comes to offspring birth weight. So in 2019, the EGG Consortium updated their GWAS analysis, doing both a fetal GWAS, so that is an individual's own genotype and their own birth weight, and a maternal GWAS, so that's mom's genotype and their offspring birth weight. So this is what we call our Miami plot. So similar to the Manhattan plot, each dot on this plot represents a genetic variant, and the x-axis is where those genetic variants are placed on the chromosome. On the y-axis here, we still have the minus log 10 p-value, but here we can see they're going up for the fetal GWAS, and it's going down for the maternal GWAS. So we do see here that there is obviously a large overlap in genetic variants that are strongly associated with both an individual's own birth weight and maternal, looking at offspring birth weight. But there is also seemingly distinct signals that are important or more important in either mom or the offspring. So Nicole Warrington took this analysis a bit further and published a paper where she used structural equation modeling to estimate the maternal and fetal specific effects on birth weight. So in this plot here, we can see on the x-axis the child effect or the fetal effect of birth weight, and the maternal effect is on the y-axis. So here we can see that there is quite a significant proportion of variants that are associated with birth weight in a positive manner for the child, whereas it is negatively associated with maternal and offspring birth weight. Additionally, at the top of this y-axis, we do see some genetic variants that seem to have a slightly stronger effect when present in the mom compared to the offspring. And just to say that those genes that we see that have a negative effect on mom and a positive effect in the child are all sort of known diabetes genes. So again, this also plays into this fetal insulin hypothesis that there is a sort of competing interests as well between mom and child. So just back to this 2016 genetic correlation plot. So this was done using an individual's own genotype and their own birth weight, where we saw this strong negative correlation between cardiovascular and glucose-related traits. This was updated in the 2019 paper. This is a little bit of a busy slide, so I'll try to talk you through it. So here on the x-axis again is all the different phenotypes they looked at and their correlation with birth weight. We have the genetic correlation coefficient on the y-axis. And we have in the circles is the fetal genetic effect. So that is the offspring genetics on birth weight adjusted for maternal genotype, and oppositely in the triangle is the maternal effect. So that's the maternal genotype on offspring birth weight adjusted for offspring genotype. And so again, I just want to highlight those glucose-related traits here. Wish I had a pointer that worked. Maybe I do. Oh yeah, there it is, sorry. So that's right here. And we could see that for the maternal effect on birth weight, there seemed to be a slight positive correlation. But when we look at the fetal effects on birth weight and the later life trait, there's a strong negative correlation as well. Lastly, from this paper, I want to just talk a little bit about an analysis they did where they estimate the causal effect of fasting glucose on offspring birth weight. So they did this two ways. First, they looked at, they used association between maternal genotype and offspring birth weight unconditional on the fetal genetics, and likewise the fetal effect on birth weight unconditional. They saw here that there seemed to be a causal effect of maternal glucose on birth weight, but they did not see an effect of fetal glucose on their own birth weight. However, when they used the adjusted effect, so that's maternal effect on birth weight adjusted for fetal genotype and fetal genetic effect on birth weight adjusted for maternal genotype, they not only saw a much stronger effect of maternal glucose on offspring birth weight, but they did see a strong negative effect of fetal glucose on their own birth weight. So over to some work that I've been doing in the Mendelian randomization space. So here we wanted to investigate whether this in utero growth restriction causes future cardiometabolic disease, and we did this in a one-sample Mendelian randomization approach. So our aim here was to investigate whether there is a causal effect of maternal intrauterine growth restriction proxied by maternal genetic variants that influences offspring birth weight on offspring later life outcomes. We looked at things like blood pressure, non-fasting glucose, body mass index, and lipid levels. We used 26,000 mother-offspring pairs and 20,000 father-offspring pairs from the Norwegian Hunt cohort, and we used genetic risk scores of maternal birth weight SNP as our exposure. For those of you that probably are not familiar with the Hunt cohort is a big population cohort from the middle of Norway, approximately. It's been collected now in 4 waves, approximately every 10 years, and every individuals in this particular county of Norway is invited to participate. The surveys generally have quite high participation rate, started at 89% for the first Hunt cohort and has now been 54% for the last 2 waves. And when we did this study, they had approximately genotyped 90% of Hunt 2 and 3. So that was the sample that we used. And in this 70,000 individuals that have genotype, there is about 15,000 siblings and 45,000 parent-offspring relationship, including those 26,000 mother-offspring pairs that we used for our main analysis. So this was our study design. Sorry, this is a very busy slide, and I'll try to talk you through it. So what we were interested in was to see if this maternal genetics that we know operates through the intrauterine environment and causes offspring birth weight, if these also had an effect on offspring cardiometabolic disease. So we used mother-offspring pairs, which allowed us to adjust for offspring genetics and then blocking this path here, meaning that if we saw an effect of maternal genetic on offspring disease, that would have to either be through the intrauterine environment here, or alternatively, maybe less likely, it could be that these genes influenced birth weight and that birth weight itself had some sort of causal effect postnatally. We also used both maternal and paternal genetics because we thought that there might be a chance that there was a postnatal effect, and then we would see that effect equally in both moms and dads. And lastly, we also just looked at the offspring genetics to see if we could find this relationship between their own genes and risk of cardiometabolic disease, so here blocking this path through the maternal genetics. So when we did all of this and looked at the maternal genetic risk score and offspring outcome in our 26,000 mother-offspring pairs, we found no evidence of any effect of birth weight on any of these later life traits that we did look at. Likewise, for in the paternal analysis, we did not find any effect on any of the offspring outcomes that we looked at. However, when we looked at using offspring genetics and offspring later life disease, we did see an effect both on systolic blood pressure and glucose, suggesting that some of this observed relationship that we see between birth weight and later life disease is mediated through the offspring's own genome. So just to switch gears a little bit, I'll talk about some of the work that we've been doing using two-sample Mendelian randomization. So firstly, I'd like to talk a little bit about this paper we did where we wanted to investigate the observed association between B12 and folate and birth weight to see if it was causal and whether or not it was mediated through maternal or fetal genome. We chose folate here because there is randomized control data on folate supplements and birth weight, so we thought that we could use this as sort of a proof of concept paper to see if this method actually worked when we wanted to look at these sort of questions. And so what we did find was that there wasn't any evidence of a causal effect of maternal or fetal B12 on the offspring birth weight, but we did find a positive causal effect of maternal folate levels on offspring birth weight corresponding to an increase in birth weight of about 71 grams per 1 standard deviation higher folate. And we did find some evidence of a small inverse effect of fetal folate levels on their own birth weight. And these results were actually consistent with evidence from randomized controlled trials that higher maternal folate levels increases offspring birth weight, suggesting that we can actually use this method when wanting to look at causal relationship between exposures in utero and offspring outcomes, which was really exciting. And so I also just want to talk about a somewhat work in progress paper that we've been working on looking at the causal effect of maternal glucose on offspring later life cardiometabolic traits. So the aim here is to investigate whether maternal fasting glucose or maternal risk of type 2 diabetes have a causal effect on offspring cardiometabolic traits such as BMI, hip to waist ratio, blood glucose, glucose measurements, lipids, and CRP. And so we did these analyses in the Norwegian Hunt cohort, UK Biobac, and Alsback cohort, and this work was led by Tom Bond and Lakshmi Bratha. And so here we have an overview of all the traits that we've looked at down on the left-hand side. We have two sets of estimates, so the glucose and maternal marginal estimate, so that's just maternal genotype on offspring traits, and we also have the conditional effect estimate, so that's when we've accounted for offspring genotype. We used birth weight as a positive control here because we know maternal glucose should have an effect on offspring birth weight, which we also saw in these analyses. Interestingly, we did see an effect of maternal glucose on offspring glucose and HbA1c when we didn't adjust for offspring genotype, but when we did that, that effect went away, suggesting again that this observed relationship might be a product of genetic pleiotropy through the offspring's genome. So leaving Mendelian randomization, I'll talk about another method that we have been using to also look into this DOHAD question. So that's genomic SEM, and I'll talk about one paper where we use genomic SEM to partition the correlation between birth weight and later life traits into maternal and fetal components. So genomic SEM is a flexible multivariate statistical method that uses GWAS summary result statistics on several different traits. It takes into account the unknown degree of overlap between our GWAS samples, and it can be used to partition trait heritability and genetic correlations into maternal and fetal components. And so it can be used to investigate genetic correlation between birth weight and later life cardiometabolic outcomes. So I'll go through our model, which is quite a busy model. Bear with me, I'll try to explain it as best as I can. So here in the bottom, we have our 4 GWAS summary statistics. So we did this analysis for many later life outcome traits, so I've just used systolic blood pressure here as an example. So we input 4 sets of GWAS summary result statistics, one which is own birth weight, so that is own genotype and own phenotype. We use offspring birth weight, so that's maternal genotype and offspring birth weight. And likewise, for our later life traits, we have own genotype and own systolic blood pressure and maternal genotype and offspring systolic blood pressure. So what we do is that we load these GWASs onto these latent variables, so own birth weight to own birth weight latent variable with corresponding with 1 here and 0.5 to the maternal GWAS due to the mums and offspring genotype being correlated with 0.5. And likewise here for offspring birth weight latent variable, that is loaded with 1 from the offspring birth weight GWAS and 0.5 from own birth weight GWAS. And similarly, we do that with the later life traits. For all of these latent variables, we can both estimate their variance and covariance between each other. So the variance here corresponds to the heritability of the trait, and the covariances is how they covariance between each other. And so what we were interested in mostly is this covariance here between the maternal genome affecting both offspring birth weight and offspring or own systolic blood pressure. So this covariance here between offspring birth weight and own systolic blood pressure, and here between offspring systolic blood pressure and offspring birth weight. Additionally, we were also interested in this covariance between own birth weight and own systolic blood pressure, so that will correspond to the genetic pleiotropy through the offspring genome. When we looked at all of these covariances, we did see a significant covariance between own birth weight and own glucose, again suggesting genetic pleiotropy through the offspring genome and corresponding very well with the results that we have gotten using other methods. We did also see a covariance between offspring birth weight and own systolic blood pressure, so that would be consistent with a causal effect of maternal later life trait on offspring birth weight. I think this observation here is a little bit more debated, where some studies have found that there is a causal effect of maternal blood pressure on birth weight and others have not. But a big question is birth weight the same as low birth weight? So in the Barker hypothesis and others, they look at low birth weight as being either, could be due to experiencing famine, low maternal nutrition, some sort of poor environment, whereas the birth weight that we tend to use are more birth weight within the normal range or birth weight from healthy pregnancies. So we did a paper that was just published very recently where we asked the question, is the genetic etiology of very low or very high birth weight the same as birth weight within the normal range? And studies like the Egg Consortium birth weight GWAS had either restricted inclusion on gestational age or birth weight above or below 2.5 and 4.5 kilos. So when we use these genetic variants for birth weight in our analysis, we are essentially testing birth weight that are within sort of normal limits, and that might not be the same as what the Doe-Had hypothesis or the Barker hypothesis really is all about. And so if the etiology is not the same, can we find any genetic variants specifically for either high or low birth weight that we could use to investigate Doe-Had? So we did all of these analyses in UK Biobank, which is a population-based cohort with both genotype information and self-reported birth weight information. There's unfortunately no data on gestational age in UK Biobank, which makes these analyses slightly more difficult, but we did the best that we could. We included white British individuals in these analyses that were not part of multiple birth. And we ran a total of 5 GWAS analyses, 1 case control of low birth weight versus normal birth weight, and high birth weight versus normal birth weight, where we set low birth weight to be those less than 2.5 kilos and high birth weight as those being more than 4.5 kilos. We also did 3 continuous birth weights, GWASs, 1 including everyone, so we didn't restrict on any birth weight measurement. We did 1 to mimic those that was done previously, so excluding everyone that was less than 2.5 or more than 4.5 kilos. And lastly, we also ran 1 analysis where everyone of less than 2.5 or more than 4.5 kilos were winsorized to the values of 2.5 and 4.5. We also ran some bivariate LD score regression analysis to estimate genetic correlation between these extremes of birth weight and cardiometabolic traits. So we did actually find that variant at 14 loci reached genome-wide significant across the 2 case-controlled GWASs. But the SNPs at all but one of these loci in our case-control analyses have previously been associated with birth weight. So it didn't seem like this decompensation of phenotypes added anything in terms of locus discovery. Another point to bear in mind is that one of the reasons for excluding these extremes of birth weight have been to avoid detection of loci that are primarily associated with gestational age rather than birth weight. So in the case of our case-controlled GWASs, we found that 3 of our genetic variants had normally association with maternal or fetal gestational age. But whilst our analysis identified a handful of loci whose primary effect were likely through gestational age, most of these loci probably had their main association with birth weight. So going back to one of these plots of genetic correlation, we have all of our traits that we looked at here on the x-axis and the correlation coefficient here at the y-axis. So here we have the low birth weight represented by the circles and our high birth weight case control represented by these triangles. And so the higher the correlation were between our high birth weight case control GWAS and the traits, you can see them going both darker red and further up on this y-axis, and likewise the lower the correlation, the further down, and also the more blue these become. And so just first here looking at the birth weight traits, so we have the GWAS that was done by the Egg Consortium. We have our birth weight in UK Biobank including absolutely everyone, our birth weight mimicking the Egg Consortium, so individuals between 2.5 and 4.5 kilos, and our winsorized birth weights. And so for the high birth weight case control GWAS, it seemed that the genetic correlation was almost 1 with all of these continuous GWASs, whereas for a low birth weight case control GWAS, it wasn't as strongly associated in the negative direction. It was between 0.7 and 0.8, suggesting that there is potentially something else going on here. These two here represent the correlation with gestational age, and we did see a slight correlation between high birth weight and gestational age, potentially a bit stronger between low birth weight and gestational age. And again, if we look here at glucose, there seems to be somewhat of a correlation between low birth weight and glucose, and a negative for high birth weight and glucose, although I should say that our study here is quite underpowered compared to those who've done with the Big Egg Consortium GWAS, so it's not as clear in our analysis here. But just to answer our own question, is the genetic etiology of very low or very high birth weight the same as birth weight within the normal range? Our genetic correlation analysis between our continuous GWASs and low birth weight GWAS suggest that there could be some other influences, but this was not seen for high birth weight, and we couldn't really pinpoint any specific genetic variants that would be specific for low birth weight. And so we did not find any strong variants that we potentially could use in a Mendelian randomization context. I should also say that there is other non-genetic methods one can use to investigate these questions, and one of the things that we do want to do is investigate whether famine during pregnancy exerts long-term effects by using regression discontinuity design. So essentially here we would have birth weight or later life trait as our outcome variable. Time here would be our assignment variable, and there would be a sharp abruption here in time where things changed, and then we would not expect other confounders to be different between these two times. So for instance, that could be introducing seasoning of rationing during World War or something like this. That would be quite a sharp introduction. And we will try to do this in the UK Biobank to see if individuals who are born on either side of this introducing or seasoning of rationing will have offspring of different birth weights, to try to look at this across 2 generations, and also look at individuals' own birth weight, because we do expect those born during rationing might have a different birth weight to those who didn't. And so just to summarize, observational studies do suggest that there is a relationship between low birth weight and later life disease. Our genetic studies suggest that this might be due to genetic correlation, and our Mendelian randomization analysis suggests that this is due to pleiotropy through the offspring genome. But we can also use other epidemiological methods, such as the regression discontinuity design, to test causality, to see whether specific environmental exposure might be different than these birth weights that are more in the normal setting. So with that, I'd like to say thank you for your attention, and if you have any questions.